This is going to be a remote learning on the integument system. What we're going to talk about first is the fact that the integument is the body's largest organ. When we're looking at the integument system, we're going to see that it provides for protection, specifically when we look at the covering of the epithelial cells in the epidermis. We'll look also in the connective tissue where we see many of the blood vessels, which provides nutrients to the epithelial cells. The dermis also has a reticular layer that provides a lot of strength and resiliency for the skin. We'll also look at smooth muscles such as the erector pili muscle, which controls the position of the hair, and the smooth muscles located around the blood vessels, which controls the diameter, so vasodilation and vasoconstriction. There's also a nervous system application or nervous tissue. We see this in the tactile sensory cells. So with this organelle, what we see is this is the primary connection with the nervous system. It helps to support and monitor sensory receptors in the skin. It provides information about touch, pressure, temperature, and pain. So when we are looking at the integument, we're primarily going to be spending most of the time on the epidermis and the dermis. These are the two subdivisions of the integument. The integument accounts for about 7 to 8% of the body's total body weight. In the epidermis, we're going to see cells that are going to have more keratinized, stratified squamous epithelial cells. And then in the dermis, we're going to look at cells that are mostly composed of dense irregular and areolar, or what we consider to be loose connective tissues. And then the subcutaneous is also referred to as it's an accessory to the integument system, and the subcutaneous, which we call the hypodermis, has, as you can see here, the fat droplets are the areolar and adipose connective tissue. So when we look at the functions, we're going to see that first off, we're going to see protection from abrasion, fluid loss, and chemical attack. So the epidermis in particular is going to be what we consider to be selectively permeable. It can absorb chemicals and drugs, but it also is water resistant, prevention of water loss or water gain. We get water loss through a series of what's called a transepidermal water loss. So we lose about 1 pint a day of evaporation of moisture from the skin. So we are losing water through the skin. And there's two ways in which we do it. The first way is going to be insensible perspiration, which is basically the release of the water vapor from sweat glands, such as the apocrine or the sebaceous gland. So these are going to release during non-activity. These are going to be usually just released as evaporation. Especially when we're looking at the sebaceous gland, the sebaceous secretions are going to mix with the insensible perspiration, and this produces a slight acidic film. And the pH is usually around 4 to 6, and that water is going to travel up, and then with the oily mixture is going to coat the surface of the skin. And what this does is it helps decrease the transepidermal water loss on a daily basis. And then the most common way that we see water loss is going to be occurring in sensible perspiration. And that's also associated when we look at temperature regulation. So the body temperature is influenced by these capillary beds that are located and the sweat glands. So as your body heats up, the cardiovascular system will release heat, triggering the water to be released out through the different types of glands. Particularly when we're looking at the gland, you have a specific one called a sweat duct, And with this sweat duct, you can see that it's very closely associated with the arterial or the cardiovascular system. The other way that we see is we'll talk about the immune defense when we get into the layering. And the immune defense is going to be located within the epidermis, and we'll go into detail on that on the next slide. Sensory reception, I mentioned the nervous system connection is going to be the tactile cells. These are also considered to be Merkel cells. They're specialized epithelial cells that stimulate specific sensory nerve endings when they're distorted by fine touch and pressure. And you can see that they're located at the top of the dermis, in the middle of the dermis, and they're also associated with the hair follicle. And again, this is just for protection. So whenever we look at the different layers, specifically what we're going to be looking at is from deep to superficial. The first one we'll talk about is the stratum basale. It is a single layer, so when we look at this, we're primarily going to be looking at these single layered cuboidal with kind of a columnar appearance. This layer, stratum basale, is also called the stratum germinativum. They're attached to the basal lamina, which is the basement membrane of the connective tissue, and basically it separates this epidermis from the dermal layer. This is also, we see these are the beginning of the keratinocytes, and the keratinocytes are the most abundant epithelial cells. Eventually, they'll be filling with protein and migrating to the surface of the skin and utilized for protection. The keratinocyte stem cells is where we see replication or division. Anytime that the superficial cells are being lost, then new cells are going to divide and then slowly migrate up into the surface of the skin. Also, what we see in the stratum basale is going to be an organ called the melanocyte, and these produce and store pigment called melanin in response to UV rays, so ultraviolet rays predominantly from the sun. Inside of the melanocytes, they have a pigment granule, and this pigment granule is called a melanosome, and these are going to be transferred into the keratinocytes. So the pigment granule will then be transferred into at the beginning into the keratinocytes, and the pigments can have different shades of color like black, brown, yellow, brown, and basically what they're going to do is the melanosomes or the melanin is going to accumulate around the nucleus of the keratinocyte and shield the DNA within the nucleus from any UV light exposure. So this area is a highly living cellular area, and we're just trying to provide protection. The stratum spinosum, this consists of 8 to 10 layers. Once we get into the stratum spinosum, this 8 to 10 layer is going to be non-dividing keratinocytes, and they're bound together, so they're joined together through desmosome. In this layer also what we see is an epidermal dendritic cell, and this participates in the immune response. These are also called Langerhans cells, and basically any microorganism that managed to penetrate the superficial layers of the epidermis, then the epidermal dendritic cells will protect or elicit an immune response. The stratum granulosum, this is called the granular layer. So when you look at this slide, you can see that it has a grainy type of an appearance to it. The layering system is going to be shorter. This is about 3 to 5 layers of keratinocytes, and basically there's going to be 2 types of granules that are going to be located in here: the keratohyalin granule and the lamellar granule. And basically both of the granules are involved in keratinization. In keratinization, this is the beginning of the process where the keratinocytes will be filled up with keratin. And keratin is a tough, fibrous protein. It's a basic structural component of what we see in hair and nails. So at this point, as the cells are going to be filling up with keratin, they'll slowly be dying as they go up, and then they'll be utilized just for protection. The stratum lucidum, right here. This is a densely packed, it's filled with keratin, usually found in thick skin. So it's about 2 to 3 layers thick. And we see thick skin in the soles of the feet, the palms of your hand. So this just provides a little bit of an extra protection. It does have a layer that's filled with the protein elidin. So there's an extra component to thick skin. The stratum corneum, this is the exposed surface of thick and thin skin. So we find this both in thick skin and thin skin. It's about 20 to 30 layers of keratinized cells. And keratinized means at this point that the cells are dead. We refer to these keratinized cells as corneocytes. And what you can see in the picture is there's no nucleus, so we consider this to be an anucleate cell, which means it's lacking a nucleus, and then the keratin is tightly packed together. When we're looking at the migration of a keratinocyte, we start back at the stratum basale, and then from the stratum basale up to the corneum, this will be about 2 weeks. So the migration here is going to take about 2 weeks to get to the stratum corneum. I'll draw this line a little bit different. So it'll take about 2 weeks to get to the initial layer. So we're looking at 2 weeks for that lifespan. And lifespan means as soon as it enters into the stratum corneum, then the lifespan is going to be about a month from here up to the surface of the skin. And basically, once they die off, they're providing an unsuitable environment for microorganisms. As I stated earlier, this one is going to be the thick skin, and this one is going to be the thin skin. The primary layer that is different between, when we talk about thick skin, is going to be 5 layers of keratinocytes. We see these in the soles of the feet, palms of the hand. They contain sweat glands, but they don't have any hair follicles or sebaceous glands. When we look at thin skin, these are going to contain hair follicles, sebaceous glands, and sweat glands. So the palms of our hands and the soles of our feet don't have any hair. The other thing is that the thick skin has more of a protective, extra protective layer, where these cells have a little bit more filling in with proteins. And that concludes the lecture.